So thank you for having us. We're excited to present our work on distinct antibody responses in adult and pediatric COVID-19 disease. By way of background, we all know now that the SARS coronavirus, or SARS-CoV-2, is an enveloped single-stranded positive sense RNA virus. And the major structural proteins are the S protein for spike, the E for envelope, M for membrane, and N for nucleocapsid. And we now know that this is transmitted primarily via respiratory droplets or close contact with infected surfaces, and that viral entry is mediated by the spike protein and interaction with ACE2 receptors on host cells. Importantly, in responding to this virus is the immune response. Novel viruses pose a particular problem because we have a lack of pre-existing immunologic memory. And so as we respond to this virus, how the immune system responds is going to be key to disease progression and disease potentially pathology. The initial control of all primary exposures and novel viruses is going to be dependent upon the innate response. And when you have your initial viral presentation, your innate system responds appropriately and quickly, you hope, and maintain some element of control while signaling to the adaptive immune response, which is mainly comprised of T cells and B cells. Viral clearance is ultimately mediated by CD8 T cells, and antibodies are formed in the collaboration of B cells and appropriate help from helper T cells. We can form antibodies and detect antibodies that are to the major structural proteins of the virus, primarily the nucleocapsid and spike proteins. And then we see typically in a primary viral infection, the accumulation of IgM, and as class switching occurs with T cell help, we see accumulation of IgG, which then we hope persists and continues to provide prevention of infecting additional cells. And then as we get past our primary response and clear virus, we hope that these will ultimately mediate protection going forward. We know that future protection is ultimately going to be mediated by memory T cells and these antibodies that are produced, whether it be by natural infection or by the production via vaccination. We now know, and it's been well characterized both here and around the globe, that the disease manifestations of COVID-19 are highly associated with different ages. In adults, the vast majority of patients experience relatively mild disease, in which most of it can be managed as an outpatient. However, those that ultimately require hospitalization have a fair amount of morbidity and mortality. Almost 80% of patients here at Columbia required invasive mechanical ventilation, and almost 40% died in hospital. On the pediatric side, it was previously thought that the children were largely asymptomatic, and that is remained true, that disease is relatively mild or asymptomatic in children, with acute illness requiring hospitalization being relatively rare and very few deaths. However, over the last couple of months, we've really had a new appreciation for a syndrome that has now been called and characterized as the multi-inflammatory syndrome in children. This syndrome is defined by the CDC as occurring in people less than 21 years of age, with a fever for at least 24 hours or more, with systemic inflammation as evidenced by laboratory findings, multi-system organ involvement, requiring a hospitalization, and then the evidence of either a previous exposure or infection with SARS-CoV-2 by PCR or by serologic testing for antibodies. We hypothesized that the different COVID-19 clinical syndromes would be associated with distinct antibody profiles. We were fortunate enough to be able to pair multiple different phenotypes of these patients in collaboration with the Department of Pathology, the Department of Pediatrics, and the Department of Medicine. We were able to gain access to convalescent plasma donors, adults, and teenagers who had developed ARDS and ultimately required hospitalization and mechanical ventilation, and children who presented with the MIS-C syndrome. We were able to recruit 15 total plasma donors, 12 ARDS patients and 11 of the MIS-C subjects. You can see here the age ranges for the different clinical phenotypes, with obviously the children being much younger. And then we're showing here that the days post-symptom onset, as defined in the adult and in the convalescent plasma donors and the ARDS subjects, as being defined as first symptom associated with primary infection in the children, those symptoms being associated with the MIS-C syndrome, which was distinct from the presentation of the adults.
All right, hi, I'm Stuart Weisberg from the Department of Pathology. So this is just one of the assays that we did for antibodies. So the Pathology Department has established a clinical assay for SARS-CoV-2 antibodies. This is a plate-based ELISA. It's basically an indirect ELISA format where the plates are coated with viral antigen. We have plates coated with the S protein, fully intact, the trimer. This, as you know, is the spike protein that mediates interaction with the host receptors required for viral entry and is the main target of neutralizing antibodies. We also have plates coated with N protein, which is the protein involved in packaging the viral genome into the ribonucleocapsid, and this is a protein that's basically involved in viral assembly and replication. So we took the clinical assay with the help of our some pathology residents and fellows, Wendy and Sandeep, and we modified it to make it more quantitative, basically performing multiple serial dilutions of the patient plasma so that we have a sense for how much antibody is actually present in the serum, and we can get at least a semi-quantitative result. So comparing the three patient groups, the convalescent plasma donors, CPD, patients with the MIS-C syndrome, and the COVID ARDS patients, the patients in the ICU, you can see that there are some differences in the sort of quantitative levels of these anti-SARS-CoV-2 antibodies. So on the left, we have the anti-S IgG levels. And as you can see here, levels tended to be higher in adult patients with the COVID ARDS syndrome compared to the MIS-C patients and the convalescent plasma donors. The same was true for the anti-S IgA levels. But strikingly, there was some differences seen with the anti-N response. So the CPD donors and the COVID ARDS patients had similar levels of anti-N IgG, but the MIS-C patients actually, compared to both groups, had strikingly lower levels of anti-N IgG. So here we have sort of distinct levels of anti-SARS-CoV-2 antibodies that are associated with the different clinical phenotypes, with some striking differences seen in the pediatric patients with the MIS-C syndrome. Because we are comparing one group of patients that are much younger than the other group of patients, so the patients in the convalescent plasma donor study were all adults, some of them older adults, and the COVID ARDS patients also tended to be older. We ran some correlations between the antibody levels and age. So as you can see here, there really was no correlation that we were able to detect between age and the level of any of the antibodies that we were detecting. So we don't think that the differences we're seeing are really necessarily related to the age of the patient. It really is tracking with the clinical syndrome, probably more than anything else. An additional assay that we worked on with the help of the Moscona and Perotto labs is a SARS-CoV-2 neutralization assay that they developed. This assay is probably a little bit different different than some of the ones that have been reported, which really look at the effects of the antibodies simply on viral entry. In this particular assay, this assay was sort of initially designed to be able to do neutralization assays on a wide variety of highly pathogenic viruses, really in the field, under BSL-2 conditions. And the way the assay works is that after the first round of infection, you infect S protein expressing target cells with a very simple VSV pseudovirus, and the VSV it basically picks up the S protein pseudotype from the cell monolayer, and in the monolayer expressing S protein will undergo multi-cycle replication in the targets, and that is tracked using fluorescent signal and is read out on a 96 well plate fluorescent reader. One thing that was quite striking is that if you remember back, the antibody levels were very similar between the CPD patients and the MISC patients, but one thing that we noticed is that the MISC patients actually had very low levels of neutralizing antibodies, and compared to both the convalescent plasma donors and the COVID ARDS patients. So although these pediatric patients had anti-S IgG, the neutralizing activity of the antibodies was actually quite poor. And here we have quantified it both ways, just with semi-quantitative analysis of the neutralization assay, as well as just using a cutoff for the presence of neutralization activity. So one question that people have about sort of antibody levels and neutralization activity is how do they correlate with one another? So as you can see on the left here, overall, if you correlate the neutralizing activity with the anti-S IgG levels across the entire cohort of patients, there is a clear correlation there, a positive correlation between anti-S IgG and neutralizing activity. But one thing that's quite apparent looking at the correlation line is that the relationship is a bit different for the MISC patients, where the y-intercept is significantly shifted downward, suggesting that for a given level of anti-S IgG, there is less neutralizing activity. And then if you kind of quantify that by creating a ratio of neutralizing activity to anti-S IgG level, particularly with comparison to the convalescent plasma donors, that the MISC patients and actually the COVID ARDS patients have less neutralizing activity per unit, if you will, of anti-S IgG. So just summarizing the data quickly, COVID ARDS is associated with elevated levels of anti-S IgG and IgA.
Mark.
Yeah, the MISC syndrome is associated with low anti-N IgG levels, and none of these associations are really related to age. In addition, antibodies from the convalescent plasma donors and the COVID ARDS subjects do demonstrate functional neutralization, but antibodies generated during the MISC syndrome are actually predominantly non-neutralizing, and we think that's a very important point. So MISC subjects contain anti-S antibodies, but these antibodies are very poorly neutralizing. So what does that mean? So it's really unclear if this is just a general feature of the pediatric response, because we don't have convalescent pediatric patients who didn't develop this syndrome, or whether this is a feature of the MIS syndrome specifically. Another very important question is whether these non-neutralizing anti-S antibodies might actually play a role in the disease pathogenesis. In addition, we found that the MISC patients seem to have a relative lack of anti-N antibodies, and that raises some questions about whether that could indicate, perhaps in pediatric patients, that there's decreased viral replication, cellular entry and invasion, which would be associated with less N antigen exposure and antibody production. And it also raises some questions about whether testing that depends on anti-N is necessarily going to be as sensitive or accurate for pediatric patients. So thank you for your attention, and we'll take any questions. Oh, I'm sorry, we have a very important acknowledgement slide, just on the many different groups that participated in this research. From the Farber lab, the Moscona and Peroto labs, the Department of Pediatrics, pathology and medicine. Being able to put these very different patient groups together really required a huge collaborative effort between these different departments.
Okay, thank you very much. And there's a question from Robbie Gartrell.
Hi, Robin, you might be muted.
Hey, I just unmuted myself. Can you hear me?
Yep.
Hi, thank you guys. It was a great talk. I'm actually curious to answer that last question that you had about the neutralizing antibodies being pathogenic. If you may be able to get some of those blood samples from the ARMOR study that's about to, or has already been underway, given that we don't really know the age limitation of ARMOR. I'm just curious if you had any other ideas of other studies that you could correlate with.
Robin, I'm not familiar with the ARMOR study, but we are working on finding additional samples, and we also have potentially some samples from these children that are followed up pretty closely after going home with this syndrome, and so we're hoping to be able to pair some of those samples to see if this is something that is only related to the acute portion of this, or whether it persists. So we are working towards finding additional samples.
Awesome, thank you.